Hello everyone, this is Lorraine Ortega, and together with me today are Florife Maribao and Lynn Fatima Toquero. We are the seventh group and will discuss on an equally important model organism, the ASCIDIANS. ASCIDIANS belong to the kingdom Animalia, phylum Chordata, subphylum Tunicata, and class Ascidiacea. ASCIDIANS bear the name sea squirts as they spit out water when they are touched or when they are alarmed by a sudden movement. Ascidians have an outer covering called tunic, hence their name tunicates. This tough outer covering is made of polysaccharide cellulose. Ascidians are invertebrate filter feeders which mostly inhabit shallow waters with 2.5% salinity. Now, in this presentation, we'll have a discussion on the nature and characteristics of ascidians, their morphology and anatomy, reproduction, behavior, habitat, feeding ecology, and diet. We'll also examine the growth and development of ascidians from fertilization to adult stage. We'll also look into the reasons why ascidians are considered a good model organism in the understanding of biological processes and phenomena. And lastly, we'll enumerate the fields of research where ascidians are usually being studied. Let us now proceed in the first part of this presentation, the nature and characteristics of ascidians. Ascidians are generally small. Their size ranges from 0.04 to 0.4 inches only. However, some solitary species can grow up to 19.7 inches, and some colonial species can measure 9.8 feet. The largest ascidian is the Staplia cylindrica, which measures 23 feet in length and 3 inches in diameter. The smallest ascidian is Minepora pedunculata, which is 0.02 inches in diameter. Ascidians are characterized by their bright colors. Their shape can vary among families. They are spherical to irregular cylindrical. Ascidians can be found either solitary or colonial, like in the picture. Most ascidians are found in shallow waters, attached to rocks, shells, pilings, ship bottoms, mud, or sand. However, there are also species that inhabit the deep waters. Like I said, ascidians are filter feeders. Their body structure is very important for them to carry out this function. Now we go over each body part. First is the branchial sac. In the picture, you can see the round structure filling in the greatest space inside. This includes the endostyle, tentacles, neural gland, and other structure attached to it. This sac is the one responsible for filtration and respiration. Next is the pharynx. The pharynx has cilia and gill slits, which can cause a current of water inside to make filtration and respiration possible. Ascidians have two openings or siphons in their body. First is the oral siphon. It is where seawater, food particles, and oxygen are drawn into. Second is atrial siphon, where water from respiration and filtration, and also fecal matter and genital products are expelled. Other body structures of ascidians can be positioned differently among different species. Gut loop can be left, right, under, or in between the sac and the body wall, but it is always near or on the side of the branchial sac. Next is the testis and ovary. It is under or in the gut loop or on the body wall. Lastly is the heart. You can see in the picture at the bottom part, it pumps blood in one direction and then changes or reverses the direction after several minutes. Ascidians and other class under the subphylum Tunicata are called tunicates because of their outer covering called tunic. Like I mentioned, this tunic is made up of polysaccharide cellulose. The cellulose-like material is secreted by the epithelium. This tunic provides the organism the protection and support as it is composed of proteins, carbohydrates, water, and structural fibers. The tunic can be clear, like the one in the first picture, or some are brightly colored. This covering has long, thin outgrowths or chest hairs, and sometimes is covered by a dense layer of sand grains. Ascidians are distributed widely in all oceans and seas. They can be found in the intertidal zone or the abyssal depths. They inhabit the benthic zone or the bottommost part of the ocean. They are attached firmly to any hard substratum like rocks, stones, shells, or algae. Some can also attach on soft, muddy, or sandy bottoms. Ascidians in their adult stage are sessile, as their posterior end is attached to the surfaces of the substratum. However, they can slowly contract their body. They can also close their branchial siphons quickly when needed, or can open it when water is drawn into. Ascidians get their nourishment from organic particles and microorganisms in water. They do this by capturing water through their oral siphon. When water is inside, the cilia in the pharyngeal wall beats, constantly producing a water current. Planktons are trapped with a specialized mucus net that lies over the gill slits. When the nets are full of food, they roll up into a thin cord which extends into the opening of the esophagus. The stomach at the bottom, which is lined with secretory cells, stimulate extracellular digestion. After digestion, the digested food moves from the stomach to the intestine, where fecal pellets are formed, and then move to the anus, and finally is expelled through the atrial siphon. Now we'll try to understand the ontogenetic development of ascidians.
Ascidians can reproduce both asexually and sexually. Asexual reproduction involves budding. Budding occurs only in some families. After an egg has been fertilized, a tadpole larva is formed. These larvae will develop into an oozoid. An oozoid does not attain sexual maturity and reproduces only through asexual reproduction. The oozoid bears the bud primordium, which is evident on its right side. This will undergo budding and becomes the first blastozoid, which bears 2 buds and in turn will bear another 2 buds after repeated budding. These blastozoid will then form the colony. In sexual reproduction, solitary and colonial ascidians reproduce differently. In solitary species, fertilization is external. A small egg comes from the atrial siphon and a specialized membrane covering the egg acts as a lactation device. In colonial species, fertilization is internal. Eggs are formed in the oviduct or atrium, which have a special incubating pockets. Hatching occurs at the larval stage when it leaves the parent. After fertilization, ascidians start as free-living larvae called a tadpole larva. They swim for a short period of time until they find a suitable substratum on which they can attach to. The tadpole larva undergoes metamorphosis until it reaches the adult stage. We will discuss more on tadpole larvae. The tadpole larva has an oval trunk, a tail that is longer than the trunk, organs, adhesive papillae on the anterior end, and a rudiment. A tadpole larva swims using its muscular tail. It uses its ocellus and otolith, which are both brain organs, to sense light and gravity. When it reaches a suitable substratum, it secretes slime and attaches to it using its 3 adhesive pubs. In the picture, only 2 are shown. When they are finally attached, a radical metamorphosis takes place. During metamorphosis, its axial complex is absorbed. This includes the notochord, nerve cord, and the tail's associated musculature. After metamorphosis, larval organs degenerate and ascidian develops from the rudiment located in the larval trunk.
So the ascidians or sea squirt has become an important model organism for biological studies, and here's why. Firstly, ascidians have simple blueprint for chordate development. Imagine at 110 cell stage, ascidians can already carry out gastrulation. Ascidians are basically simple organisms. They are small, with about 2,600 cells, yet are diversified cells. They are nearly transparent and have relatively short life cycle and can be maintained at high density, which means a rapid development up to 18 hours from fertilization to terminal differentiation. Most importantly, they are hermaphroditic and self-fertile. These facts and its remarkable cellular simplicity greatly facilitate it being a good model organism useful for the researchers. Another reason one makes ascidian a model organism is their homology to vertebrates. Many genes present in multiple copies in vertebrate appear to have a single homologue in ascidians. Even parts of ascidians correspond to vertebrates' forebrain, midbrain, hindbrain, and spinal cord, as you can see in the picture. As model organisms, they offer having a small, compact genome. Hence, ascidian genome appears to contain the basic chordate genetic toolkit and thus represent an ideal reference point for the expanded study of vertebrate genomes. Ascidians has also this so-called cell fate restriction, which are tissue-specific enhancers that direct the expression of embryos notochord, tail muscles, and CNS, allowing easier identification of DNA regulatory elements. It unfolds a predictable program of cell division. Lastly, they possess the characteristics of being a chordate, having a notochord, dorsal hollow nerve cord, and pharynx perforations or gill slits. However, tunicates have also a highly developed blood vascular system with a heart and a nervous system that can be easily studied because of its simplicity. Hence, ascidians are considered as our nearest invertebrate relative.
The following are some researches on ascidians. First, ascidians and chordate development. Because of their simple body plan and diversified yet few cells, ascidians are used in understanding cell fate specification among chordates during embryogenesis. A research entitled Ascidians and the Plasticity of the Chordate Developmental Program is a review of the comparison of developmental strategies among ascidians and vertebrates. Authors suggest that even if adult ascidians and vertebrates vary greatly, and that same molecules are involved at morphological level. Furthermore, the very divergence on the developmental strategies point out to the plasticity of chordate development. Next is ascidians and genome analysis. So there is a research entitled The Ascidians as a Model Organism in Developmental and Evolutionary Biology. It is because the simplicity of the ascidian genome is a key in studying gene function. These organisms are suitable for gene analysis because they have a short life cycle for about 1 to 2 months. And these organisms are small and have sperm that can be cryopreserved and are hermaphroditic and self-fertile. This cellular simplicity and rapid development from fertilization and terminal differentiation for 18 hours allows rapid screening for mutant phenotypes at single-cell resolution.
Ascidians as environment indicator. Pollutants have been found to negatively affect the immune responses of tunicates, specifically phagocytosis and cellular cytotoxicity. Hutchinson and Raftus found that tributyltin, or TBT, and copper, which are common anti-fouling agents used by boaters in the ocean to keep sessile invertebrates off their hulls, and creosote, which is used on wharves and pilings, causes the immune responses of tunicates to be reduced and even lethal. Tunicates are particularly susceptible to pollutants because of their filter-feeding lifestyle and the layer of endothelial cells that cover the pharynx. TBT is now banned from most areas and was replaced with a copper-based agent which still remains to cause harm to the tunicates.
Ascidians in drug development. Ascidians are invertebrate chordates and prolific producers of wide variety of biologically active secondary metabolites from cyclic peptides to aromatic alkaloids. Several of these compounds have properties which make them candidate for potential new drug to treat diseases such as cancer. Many of these natural products are not produced by the ascidians themselves, rather by their associated symbionts. A review by Waters D. focused mainly on the mechanism of action of important classes of cytotoxic molecules isolated from ascidians. These toxins affect DNA transcription, protein translation, drug efflux pumps, signaling pathways, and the cytoskeleton. Two ascidian compounds have already found application in the treatment of cancer, and others are being investigated for their potential in cancer, neurodegenerative, and other diseases. Ascidians or sea squirts are model for toxic screening. These marine animals provide a source of diversified biologically active products. The isolated compounds from ascidians hold an approved drug for anti-tumor, and several others are useful drug leads. They are also out for the study about neurodevelopmental defects, as numerous researches show that ascidians' brain formation is sensitive to virulent materials, most especially from endocrine disruptors. In the 21st century, ascidians are predicted to become an important source of marine blue technology.